Hello, everybody. You have successfully found the remote lecture. So let's get started. Before I jump right into the material, there was one aspect of the course design that I didn't mention actually to either group in the morning. So I just wanted to take a moment to draw your attention to one other feature that I'm hoping will help some of you bridge that gap from an undergraduate learner to a graduate learner. I mentioned to both groups that this course is going to challenge you to think on a deeper level and to be able to make connections between material, to critically think about information rather than regurgitate facts. Um, and that can be, as I mentioned, a struggle for some students to make that leap. Like, I've always memorized my notes. Like, I don't know how to think different if this is the way I've always done it. And because we only have a few exams in the class, I didn't want anyone to feel surprised by the depth of any questions on the exam. So I wanted to give you the opportunity to kind of get inside my head as you're studying to kind of test yourself and see if you're thinking about things to the right depth. So let me just jump off this screen and show you something on your D2L page. So on your main home screen, and your screen might look a little different than mine, there's always news. So if I scroll down a little bit, this is what the news has been for you. There was a little welcome to the spring, and I told you about the book. Also mentioned the paper notes in the news in case you didn't see it there. But every week, there'll be a new piece of news, and it will be a practice short answer question. The exam that you're going to take is going to be multiple choice questions, but it can be helpful for you to think about a concept on your own and see if you can draw the most important aspects of what the question is asking and put it down in a written answer. So I really encourage you to try these, to study the material from the week, close your notes, read this question, and from your memory of your notes, write the answer out. You could verbally tell a friend if that's more your thing, but I really recommend writing it because if you actually commit to what you put down on paper, and then I will post what I consider to be a well-rounded, kind of quote unquote good answer to this question. And then you can compare yours to that and see if you had the depth, if you made the connections. One thing I really want you to draw attention to is in your answer, did you only include what was relevant and important? Or did you write everything you knew about this topic? What I find a lot of times students have a hard time prioritizing. If I told you all the steps of cellular injury, but then I asked you a specific thing about one of those steps, I find that students have a hard time being able to identify which part of that huge big process is the most important for this very specific question. So make your answer as specific as you can, but include as much depth to fully support your answer. And then check it. And we'll have these every week so that you can get a gauge on your depth of studying. And if my answers are in a greater detail than what you had down on paper, or it was very different from your answer, that should help guide you to tweak your studying. If you feel maybe less of a pump up, maybe that felt more like being punched, but hopefully it was a little bit of a motivation. I am coming right off of a workout before doing this lecture, which gets me super pumped up and excited to do this, which also I just highly recommend. If any of you guys weren't thinking about exercising today, man, try to get something in. Even if you pause this video at some moment and do like 10 jumping jacks or 10 burpees, get your heart rate up. Man, you get a little hit of your natural high and it'll push you through a boring lecture, which I guess means you should do it for someone else's lecture because clearly this stuff isn't boring. We're talking about cells getting hurt. Let's jump right into it. All right, both groups ended here. We are talking about the types of tissues that are either vulnerable or resistant to ischemic injury. And it really comes down to the type of cells that are there, right? Cells that are more metabolically active are going to require more oxygen and nutrients. Quiescent tissue, less affected. If that tissue has a stored source of glycogen, it's got glucose right where it needs it, and it's going to be able to run anaerobic metabolism more effective and for a longer duration than cells that do not store glucose. So kind of tying the concepts of ATP depletion and the reliance on anaerobic metabolism to what we see clinically in the tissue types that get more injured from ischemic injury and less injured. Okay, so hopefully we've laid the groundwork in lecture 1 that when you do not have sufficient oxygen, you will not be able to keep up with ATP supply. ATP will become depleted in a state of hypoxia. Now, you have this figure here in your notes for you to draw right on if you'd like. I prefer to draw kind of on a whiteboard in my cartoon form, so I'm actually going to redraw this cell, and you can either choose to redraw it or just add to the kind of empty diagram that's in your notes. All right, so we're drawing our generic cell, just like you have there in your notes. And let's just kind of remember the resting kind of life of a cell. The cell maintains a resting membrane potential, meaning it keeps kind of a difference in its charge because it actively moves ions in and out. And one of the most important transporters in all of physiology is the sodium-potassium exchange. So this is an active transport, so it requires ATP, and this transporter pumps out 3 sodium ions and it trades in for 2 potassium ions. And this is relevant to our story because in a healthy normal cell, you have way more sodium outside of the cell than inside of the cell. There's tons of it out here. We'll learn the concentrations of sodium in our lab values, but we're looking at something like 140 milliequivalents per liter, typically in extracellular fluid for sodium, with very little in the intracellular. There's a tiny bit, but if you compare it, it's about 12 milliequivalents. Again, for a studying reference, these aren't numbers I would ever put in a multiple choice question, but it helps to illustrate our story if we know the values for these numbers. So don't store them in your brain, but appreciate the story. All right, so there's way more sodium outside of the cell than inside because of the pumping action of the sodium-potassium exchanger. Now, we are experiencing hypoxia. So this is now going to become a hypoxic cell. And hypoxia means reduced amounts of oxygen. And we know that if the cell can't make enough oxygen, it's going to deplete its ATP. So if this cell does not have sufficient ATP, it is going to be failing to pump out the sodium and pump in the potassium. Let me say that one more time. If this cell does not have sufficient ATP, you shut off the sodium-potassium exchanger, which means you will be no longer able to pump out that excessive amount of sodium. So if you're not pumping the sodium out, and there is way more sodium outside of the cell than inside, in other words, we have this huge concentration gradient of sodium outside of the cell compared to inside of the cell, what is going to naturally happen? When you have a bunch of sodium on the outside, very little on the inside, this sodium is going to leak into the cell. So with ATP depletion, sodium is going to leak into the cell and raise the intracellular sodium concentration. Now, in physiology, an important relationship, if it hasn't already been strongly established in your mind, it's important to know that in physiology, water follows sodium. This is just a really important concept. If you want to dust off some physiology cobwebs, this is one of the main mechanisms whereby we can concentrate or dilute our urine in the kidney. If we manipulate where the sodium goes, water follows sodium. So by manipulating where sodium goes, we can kind of direct where the water goes. That's an example of a regulated use of using sodium to move water. In this case, we are experiencing hypoxia. The sodium is leaking in, kind of contrary to what we would quote unquote want for the cell, and water follows sodium. So if sodium leaks in, so does water. If you start having a bunch of water moving into the cell, what would be the consequence of that? Increased intracellular water is going to cause this cell to swell. So I'm just expanding. If you want to put arrows in your notes so you can tell this is new, the cell membrane has been pushed outward because the cell is now swollen with water. So the reason the cell is swollen is because without ATP, you cannot maintain normal ionic gradients across the cellular membrane. When you can't pump the sodium out, it will leak in and the water follows sodium. So now we have this swollen cell kind of engorged in water, which in and of itself maybe isn't a problem, but it's going to cause problems because as the cell swells, so do the organelles within it. So the mitochondria within the cell swells. The endoplasmic reticulum within the cell swells. Let's go back to the PowerPoint to talk a little bit about this effect on the organelles. So we mentioned that the endoplasmic reticulum dilates and swells. And the endoplasmic reticulum is a really important part of the cell because it has these like ribbons of tissue spotted with ribosomes. That's why it's the rough endoplasmic reticulum. All those little bumps are ribosomes. And ribosomes are the little kind of, maybe think of them as little protein factories. They are where we start putting the structure of the proteins together to build brand new proteins. So if the endoplasmic reticulum is getting swollen and dilated, it causes ribosome detachment. So these little ribosomes, these little protein factories, start drifting away from the endoplasmic reticulum. Well, if they drift away from the endoplasmic reticulum, this is going to reduce protein synthesis because that's the site of where proteins are built. So as a result of the cell swelling, we actually get worse at building new proteins. And remember, our proteins are always in flux. We're breaking them down and we're building them. And we're injured, so we might be wanting to build some proteins to help during this, state of stress. But the state of stress is actually making us worse at making proteins. Okay, so this falls under the kind of overarching problem that comes with cellular swelling. So we're going to go through the mechanisms of cell injury. If you remember, we saw this slide on Wednesday, and we've discussed and gone through what happens with the depletion of ATP. Let's now go to step 2 and talk about what happens when the next step results in calcium moving into the cell. So again, you have an image very much like this in your notes that you can kind of draw on and add to as we discuss this next phenomenon. So this kind of step 2 of cellular injury is a loss of intracellular calcium homeostasis. Let's just take a step back before we talk about what happens to make sure everybody knows how important calcium is in the cell. There is almost no calcium in the intracellular fluid of the cell on the inside. That's why I made this guy so tiny. We are in the nanomolar range of calcium in the intracellular fluid. And we maintain almost no calcium in the cytosol because calcium is this incredibly important intracellular signal. You could think of calcium as almost like a light switch. And because there's almost no calcium inside of the cell, that means that the amount of calcium outside of the cell is incredibly larger. We're talking about almost a 10,000-fold difference in the amount of calcium outside of the cell compared to the amount of calcium inside the cell. So I said calcium is kind of like a light switch, because if the cell wants to very quickly send a signal to let some process inside of the cell know it has to happen, it can open a calcium channel for the briefest of brief moments, and calcium will flood into the cell because there is this huge concentration difference. This is normal physiology. And then it can shut the gate and instantly pump all that calcium back out, and you turn the light switch back off. So in normal physiology, the cell maintains these huge differences in calcium gradients to allow calcium to be this incredibly potent intracellular signal. So the ability to quickly move the calcium in is helpful, but then obviously you have to pump it back out. A calcium gradient like this isn't maintained naturally, and this gradient of calcium requires ATP. And it's drawn into your diagram here. You can see at the endoplasmic reticulum here, you have an active transporter that actually pumps calcium into the ER. You have an active transporter that pumps the calcium out of the cell. So it requires a lot of energy to maintain this huge concentration gradient. So in step 1 of the cellular injury, we depleted our ATP, which means we are not going to have enough ATP to actively pump the calcium out of the intracellular fluid. What will be the consequence of that? Calcium is going to enter the cell, a huge concentration gradient. So it's a very similar mechanism to the story with sodium, except the gradient here is much, much more significant. And the role of calcium inside of the cell is more critical to the life of the cell because it's that intracellular signal. So I mentioned that you could think of calcium like a light switch, but I didn't really tell you what lamp was being turned on by that light switch. The light switch that the calcium turns on is it regulates enzymatic activity, among many other things. But let's talk about enzymes for our story. When you raise intracellular calcium concentrations, you turn on a certain group of enzymes. Now, if you're kind of following along in your notes, kind of point A under this lost intracellular calcium right below the diagram, it says that we've elevated the intracellular calcium concentration, and then it says you have uncontrolled activation of cellular enzymes. And the word uncontrolled is incredibly important there, because in normal physiology, we open calcium channels, let the calcium levels inside of the cell spike in order to turn these on, but then we quickly turn them back off again. Because there are times when each of the enzymes we're about to discuss may have a good job to do in the healthy life of the cell. But in a state of cellular injury, the calcium is flooding in abnormally. The intracellular calcium concentration is high in a pathological way, not in a regulated physiological way. So because of that, we don't have a regulated activation of enzymes. We have uncontrolled activation of enzymes. We're turning every light switch on, and whatever your job is, do it now. That's basically what the calcium is saying. So let's look at the enzymes that are going to get turned on in this process. Now, if you're less familiar with chemistry and enzymes and kind of biochem, there's some shortcuts here for studying. So the enzymes end in ASE. So So that already helps you know we were talking about an enzyme. So all the ACEs are enzymes. And then the first half in these enzymes actually tell you what they break down. Okay? So look at the first one, phospholipase. Okay, so I have a lipase. This is going to be an enzyme because it ends in ACE. What does this enzyme break down? Phospholipase. What does that sound like? Phospholipases break down phospholipids. Okay, phospholipids. So in cellular injury, we have uncontrolled activation of phospholipase. So let's start breaking down phospholipids. Okay, so the next question you have to ask, okay, if they're breaking down phospholipids, where are phospholipids? Why do I care about that? Does that sound familiar at all? Phospholipids. Where are those found in the cell? If you're listening with somebody else, pause the video and tell each other where you're going to find it. All right, welcome back. I hope you all paused the video and thought about it for yourself. Where did you find it? In the cell membrane. That's right. The bilayer, phospholipid bilayer of the cell membrane. Excellent job, you guys. Okay, so now we have uncontrolled activation of an enzyme that is going to start breaking down the phospholipids of the cell membrane. That does not sound good. I'm just saying. In my opinion, that does not sound good. Okay, let's go on to the next one. Protease. Okay, ACE tells me it's an enzyme. What is it breaking down? Protease. What does that sound like? It's breaking down proteins. Okay, so now we have uncontrolled activation of an enzyme that's going to break down our proteins. Again, always ask yourself, though, why do I care about this? And hopefully you care a little bit because you have to learn this. But as from the cellular perspective, why do you care about it? Where are proteins found? Well, we have a intricate cytoskeleton within a cell. Does that sound familiar? The cytoskeleton is a network of interlinking filaments, basically long chains of proteins, and they attach to the membrane, and they really give the cell its shape and its structure, right? They kind of hold it all in place. So proteases are going to kind of non-discriminately start to degrade and break down proteins, which means during cellular injury, we are going to start breaking down the cytoskeleton. Cytoskeleton is going to detach from the membrane. So normally it's anchored to the membrane, which is what holds kind of the reason we draw a cell often like a nice little circle, even though they're not all circles, is because the cytoskeleton actually creates a nice shape for our cells. So now we're going to have cytoskeleton detachment, cytoskeleton breakdown. Another important place that we have proteins in the cell is we actually have proteins embedded in the cellular membrane. Let me show you a picture of this. So here you can see the phospholipid bilayer, right? So where'd my pen go? This is the cell membrane. So there are those phospholipids. And this is the cytoskeleton, the long chains of proteins that proteases are kind of chewing up and degrading. But you also have these proteins that are embedded within the membrane. So of our first two enzymes that we spoke about, phospholipase and proteases, we have two enzymes that are actively degrading elements of our cellular membrane. This is really important because the cell membrane, keeps the intracellular environment separate and protected from the extracellular environment. Once you disrupt the membrane, you're really going to lose any control you have over the elements within the cell. So because of the uncontrolled activation of enzymes, because of high intracellular calcium, we now have an unregulated increased activity of phospholipase and proteases attacking the lipids of the membrane and the proteins of the membrane and the cytoskeleton that attaches it all together. The third enzyme that gets turned on in our story is endonuclease. So endonuclease, again, tells you it's an enzyme, and the nuclei in the middle tells you that it separates nucleotides, excuse me. So endonuclease, if it disrupts nucleotides, degrades DNA. So endonuclease is going to start to break down and degrade the DNA in the cell. Again, that does not sound good, right? DNA, let's put it in the camp of things that are important to the cell. All right, the last one, ATPase. Again, the name tells you what it breaks down. ACE, it's an enzyme. What does it break down? It breaks down ATP. Our story started with ATP depletion. Insufficient ATP caused this whole problem. And now we're turning on an enzyme that's going to further break down any ATP that we have. This is what we call in physiology a vicious cycle. There is a change that happens that initiates a series of events that ultimately makes the initial stimulus even worse. A loss of ATP in the cell creates an environment where we lose control of our ionic gradients, which results in an unregulated activation of enzymes, which ultimately further deplete ATP. And around we go. That's a vicious cycle. I included in your notes just this picture where you can see the difference between, let's say, happy cell and injured cell. And you can see the happy cell has a very nice shape to it, a distinct shape, where the injured cell has lost that integrity. So due to the swelling, because of the sodium that moved in the water that followed it, the cell has gotten larger and it's pushing on its barrier, the cell membrane. Then you've lost the integrity of the structural elements of the cell because the cytoskeleton has been degraded. And so instead of maintaining its nice round shape, it starts just to kind of squish out and create these little bulbous protrusions. And these little saccule things that are poking off are called blebs. So this phenomenon is known as cellular blebbing because the lack of cytoskeleton and the breakdown of the phospholipids as the water inside of the cell is pushing out allows the cell membrane to kind of bulge out in these little pocketed forms. Actually, here you can even see some rupture where the cell membrane is actually ruptured open because the phospholipids and the proteins have been so degraded. And this is the cell is on its way to being further injured, especially if we have any kind of rupture happen, then we're moving into irreversible injury. But the blebbing happens as the cytoskeleton has detached, the water is pushing out, and you lose the integrity of the cellular shape. And it becomes very different looking and visible under a microscope. Okay, so in the next page of your notes, I have this kind of summary because I really want to drive home that every step we've talked about so far has been a step of reversible cellular injury. So on the left in your notes, you have my cartoon version of a normal happy cell. You have a nucleus with normal looking genetic material on the inside. That's here. You have a normal looking endoplasmic reticulum with ribosomes attached, normal sized mitochondria. The cell has a nice shape. It has a nice size. It's a normal healthy cell. It's doing its job. All right. Then we have, hypoxia hits for whatever clinical reason, and hypoxia is going to cause a loss of ATP. With insufficient ATP, you are going to rely on anaerobic metabolism. If you are relying on glycolysis in the absence of oxygen, the end product is lactic acid. As you accumulate lactic acid, the cell becomes more acidic, which inhibits certain enzyme activities, and it also gives you this visual of chromatin clumping. So we can see visible evidence that the cell is experiencing stress because the nucleus looks very different under a microscope. The size of the cell has gotten much larger as sodium has leaked into the cell and water has followed. We've completely lost the shape of the cell because the influx of calcium has turned on a bunch of enzymes, including phospholipase and proteases, which have degraded the cytoskeleton, caused cytoskeleton detachment, which gives us these characteristic blebs. Oops, my blebs kind of get, this is supposed to be an E, blebs, coming out on the cell. The water that has moved in has engorged the mitochondria. This is much larger. The mitochondria, there we go. The endoplasmic reticulum has swelled, and we see ribosome detachment. Ribosomes, again, is the site of protein synthesis. So this ER is definitely not making as many proteins now that it is in this state of injury. And this looks pretty ugly. Like, this does not look good for the cell. But at this point, if you were able to reverse the hypoxia, if you were able to restore oxygen, let's say you busted the clot, you got the person on oxygen, you stopped the carbon monoxide poisoning, whatever is your cause of hypoxia, you were able to restore oxygen to the cell, this would completely go back to normal. Despite how ugly this looks, this is all reversible at this point. So, these are the steps of cellular injury that lead to reversible cellular injury. Before we go on to the progression to irreversible cell injury, I just want to take a quick spot in your notes to mention that it's not always perfect or great when you fix the hypoxia, especially when we're talking about ischemia. So ischemia is reduced blood flow to a tissue. So let's just use the example of a blood clot. A blood clot blocks the blood flow and the cell becomes ischemic. During that ischemia, the cell goes through the steps of reversible cell injury, right? The ATP depletion, the loss of intracellular calcium, the cellular swelling, the uncontrolled activation of enzymes, the cellular blebbing, the loss of structure, all those things are happening. And then you get a clot buster on board, or your body breaks down the clot, or it dislodges the clot, and suddenly you reperfuse the tissue. In some cases, the reperfusion can cause an injury. I don't want you to struggle with this too much in the terms of studying purposes, but it's important to know clinically because we often treat patients who have ischemia reperfusion injury. So when someone's ischemic, even though this is a possibility of happening, the overarching goal is restore blood flow as quickly as possible. But when you suddenly deliver a bunch of oxygen to a cell that looks like this guy here, especially with this big swollen mitochondria and abnormal organelle kind of function, the sudden introduction of oxygen can actually cause an injury. And I kind of listed the reasons that it can cause an injury. Let's just think about them for a second. This isn't a list to memorize per se, but I just want you to appreciate how an injury can happen at this point if you suddenly get oxygen. For one, the mitochondria is not happy and healthy at the time when you suddenly give it a bunch of oxygen. So if you suddenly give a kind of sick, dysfunctional mitochondria a huge supply of oxygen, it's going to start producing reactive oxygen species. So on this slide, this is ROS. ROS is reactive oxygen species. Reactive oxygen species is a name for an oxygen molecule that has an extra electron, which makes it a highly reactive molecule that can damage things within the cell. So you start producing an oxidant, basically a volatile oxygen species that can cause damage. On top of that, you had this extremely high calcium already. And if you suddenly give oxygen to the cell, this can actually cause contractures, which when we're talking about myocytes can be a huge problem because this could impair cardiac function if you suddenly are having contractures of myocytes at the point of reperfusion. The mitochondria can become further damaged because the restoration of oxygen can open what's called the mitochondrial permeability transition pore, the MPP. And this is written out in your notes for you. Basically, when the MPP opens, the mitochondria suddenly becomes much more permeable to elements in the intracellular environment, the fluid around it. The blood vessels around this cell, so I could kind of draw in a blood vessel next to it, can have endothelial dysfunction, which means the blood vessels that are right by the cell having the injury can, as it says in your notes, here's the fancy words first, and then let's break it down. Because of the endothelial dysfunction, the blood vessel right by the cell has a prothrombogenic phenotype. That is a very fancy way of saying this blood vessel right here is much more likely to form clots. Prothrombogenic phenotype basically means the environment in the blood vessel wants to form clots. So you just fixed the blood flow problem, but the sudden restoration of oxygen can damage the endothelial cells, making a new clot form. You can also get an inflammatory response in the cell, which can cause edema, and we'll kind of go through some of those steps. So try not to get too dragged down in those details. I know it's a little heavy, but I wanted to have it in your notes at the point where we're talking about saving these cells from ischemia, that sometimes that can actually cause further damage. And there's not really a great way of predicting, when that would happen. So it's sometimes a clinical consequence that we have to deal with. Okay, so this is again kind of that cartoony picture of our injured cell up into the point where it's still reversible. At this point, though, we're going to continue our story when we don't restore oxygen. We haven't fixed the blood flow problem. We haven't gotten our patient on oxygen or whatever is the cause of the hypoxia. And this cell is going to continue in a state of hypoxia. So that we're still moving down the same steps, but now we've reached kind of that critical junction where from now on, things become irreversible. So even if they get to the hospital and they bust the clot, these cells aren't savable anymore. They have become too injured once these next steps happen. So for your studying purposes, if you look at your notes on the bottom of page 8, I rewrote the steps from the beginning because I don't want you to go to this section of your notes when you're studying and think think this is step 1 and forget that this stuff already happened. So 1 and 2 of irreversible cell injury is the same as what we just talked about, right? ATP depletion occurs. There's a reliance on anaerobic metabolism, but it will be insufficient to maintain ATP, which is going to result in a loss of your ability to maintain your sodium gradients, which is going to cause cellular swelling. You will lose your calcium homeostasis, again, because of the loss of ATP. So your enzymes are still turned on, right? You still have phospholipase. It's just going to continue to damage the membrane. And it will continue to damage the membrane until there is disruption of the cellular membrane. So that's a key point. With phospholipase activated long enough, it will eventually break open the membrane. And at that point, intracellular elements are going to leak out of the cell. The phospholipase, one of the membranes we didn't really talk about in reversible cellular injury is the fact that within a cell we have lysosomes. We mentioned this with autophagy, We talked about how the cell can eat itself. A lysosome is like a little packet of digestive enzymes that are, will just break down proteins, but it's held together by a phospholipid bilayer within the cell, so it's protected. So it has this membrane. But phospholipase is also going to break down the membrane of our lysosomes, which float around inside of the cell. That is a terrible thing for a cell to have happen if it plans to live, because once the digestive enzymes of the lysosome are exposed to the cellular elements, they're just going to break them down and start eating them without control. So in irreversible cell injury, phospholipase has longer to work, which gives it time to fully break through the membrane or even break through lysosome membranes. Proteases will continue to break through and break down proteins. Endonuclease, right? That's the one that's attaching the DNA. This will cause DNA damage to the point where it will eventually cause cell death. Your DNA can only withstand so much damage before the cell will not continue living, because your DNA is its blueprint, and without its blueprint, it will not be able to sustain cellular life. So number 3, when we're actually in irreversible cell injury, is actually to talk about those reactive oxygen species again. So reactive oxygen species can be described a number of different ways. Really depends on which reactive molecule you're talking about. So in your notes, it says reactive oxygen species, ROS, accumulate / oxidative stress. On the slide here, you say accumulation of oxygen-free radicals. This is talking about the same thing. Oxygen-free radicals is reactive oxygen species. So I mentioned that oxygen is normally a stable molecule. It is not reactive, but when you either add on or remove an electron, you can create a free radical. In the case of oxygen, we call this a superoxide because it has this extra electron added. So electrons like to be in pairs, and that's what makes it a stable molecule. We call any molecule a free radical when it has a single unpaired electron in its outer orbit. And we talk a lot about free radicals as a negative thing. As I mentioned a few minutes ago, a free radical has the capacity to damage basically anything it bumps into. So if whatever's kind of adjacent to it, if it bumps into it, whether it's a protein or a lipid, a carb, a nucleic acid, it doesn't really matter. It has the chance to damage or degrade it. And this is important because at this point in irreversible cell injury, we start to accumulate these free radicals. And the most common free radical in the cell is reactive oxygen species, which is why I give you that example in your notes of how molecular oxygen can be converted into a superoxide radical. So when you take a normal oxygen molecule, which is stable, and you add on an extra electron, you get this superoxide radical because now you have an unpaired electron in its outer orbit. And this guy here can damage anything it bumps into. It's a non-discriminate damager. So in the step 2, we talked about how calcium turns on these specific enzymes, and those enzymes are like targeted attacks, right? They're like heat-seeking missiles for phospholipases and proteins. Now we're just spilling out a bunch of stuff that like whatever it touches, it damages. So it can cause similar damage, but in a less organized way. It's less effective, right, of breaking down proteins because it would break down anything, but it adds on to the damage within the cell. So where do these come from? I just mentioned that they're accumulating. Well, the mitochondria actually makes reactive oxygen species all the time in our healthy normal cells. This is a byproduct of normal aerobic respiration. So when your body is making ATP, part of that process actually makes some reactive oxygen species. So in almost everything in physiology, a little of something is good and too much is bad, but absolutely nothing is, a complete absence of it is not good either. The reactive oxygen species is actually a very important intracellular signal in a normal healthy cell. When life is good, within our cell, we have what's called antioxidants. That's a pretty common term that you've heard before. And antioxidants within the cell help to block the negative aspects of reactive oxygen species. So in a normal, healthy situation, the antioxidants balance out and keep in check the production of reactive oxygen species. In an injured cell, the mitochondria is not functioning normally, right? It is swollen. It doesn't have sufficient oxygen for normal cellular respiration. So it starts spitting out extra reactive oxygen species. Another common source of reactive oxygen species are white blood cells. We will talk about the role of white blood cells in the body next week, but they're really good. We always call them they're good eaters. They can show up and eat bacteria and eat cellular garbage and chew it up and spit it out. Well, they eat a lot of it up. I'm air quoting eat. You guys can't see me. I still act everything out even though I'm talking to a screen. They quote unquote eat everything up because they degrade and break down things. And one of their main ways of doing that is they pump out reactive oxygen species, which damage the elements that those white blood cells are trying to attack. So neutrophils, macrophages have the ability of producing reactive oxygen species. So as I mentioned, as it's written in your notes, we normally in a healthy cell keep these in balance because we have natural defenses against reactive oxygen species. So superoxide dismutase, SOD, is an enzyme that actively breaks down reactive oxygen species. That's its job. And when you have enough of those enzymes, you're very good at keeping it in check. We have antioxidants like vitamin C and E, and also what's called just natural decay. That reactive species will bounce around for a little while and it'll kind of fade out and it will be done doing its damage. It just naturally goes away. So in a normal, healthy environment, there's some degree of reactive oxygen species, but we are balanced and kept in check. The term oxidative stress suggests that there are more free radicals than we have the capacity to neutralize with these defenses against them. We are accumulating oxidative free radicals within the cell. That is the stress. The oxidative free radicals are the stress. So if you have more of these reactive molecules floating around in the cell, you are going to get damage. They're not going to pick what they damage, they're just going to damage whatever they bump into. So in your notes underneath oxidative stress, I just have some lists of places and times when we see oxidative stress. It's very common for cells to experience oxidative stress when you have an infection. Again, because those phagocytic cells, like macrophages and neutrophils, produce reactive oxygen species to actually kill off the infection. So the generation of reactive oxygen species is beneficial to kill the bacteria, but it can also end up killing normal cells as well. That's why we get into trouble when we have an infection, because our response to the infection is actually normally what causes most of our symptoms. Ionizing radiation. So UV rays is the most common source of oxidative stress in the body. UV rays is the most common source of ionizing radiation that our body experiences coming from the sun. But that ionizing radiation can increase the oxidative stress in the cells. certain chemicals can. And then we already talked about this ischemia reperfusion injury. When you suddenly give a bunch of oxygen to a dysfunctional mitochondria, it starts to pump it out. Okay, so in our story of our hypoxic cell that's getting injured, and now we're starting to accumulate reactive oxygen species, what is the effect of that? Well, the oxidative free radicals can cause protein modifications. They can cause cross-linking or fragment proteins. So this is problematic for the cell because we're further breaking down proteins. One thing to keep in mind is that enzymes are proteins. So with kind of this cross-linking and this damage or change to some of the proteins, in some cases this increases enzymatic activity, and in other cases it actually reduces enzymatic activity. So it's kind of a mixed bag. We also get lipid peroxidation. So this is creating abnormal lipids within the cell. So again, we're kind of thinking about that cellular membrane because that's where a bunch of our lipids are. So earlier in our story, we talked about phospholipase being this specific attack on the cell membrane, but now we're also having a bunch of reactive oxygen species kind of bumping into the membrane and causing further damage. Lastly, reactive oxygen species can further damage the DNA. So again, endonucleases was an enzyme that was specifically targeting the DNA. Now we have just kind of a general damage to cellular elements by reactive oxygen species that includes DNA. As the DNA starts to become damaged, you can actually see evidence of nuclear damage visible if you put a cell under a microscope. So as the DNA material within the nucleus becomes further and further damaged, you can actually see changes. So pyknosis is when you get this solid, shrunken, dark mass of genetic material within the nucleus. Then it can progress to getting these little nucleic fragments. And then finally, karyolysis is when the chromatin has completely dissolved. So you've completely lost any integrity to your DNA at all. All right, so if we were sitting in 356 right now, I would have gone about 2.5 minutes over, and you guys would be like looking at the clock, wondering if I was going to give you a break. So what I'm going to do is I'm going to end this video now and pick up on a second link. I find that shorter videos can maybe be a little more psychologically tolerable than like a 2-hour YouTube video. So I'm going to end this link here, and I will pick it up on the next link. Both of these video recordings are for week 1.